presentation. My name is Andrew Olson, and I'm the Vice President for Business Development at Advaita, and I'll be moderating today's webinar. Just as a matter of housekeeping, everybody is on mute, but you can certainly ask questions using the question box in the control panel. If anybody wanted to just give me a hi or it's working, that would be great. That way we have confirmation that things are working. And obviously, as you have questions throughout the presentation, please use the question box to just send us a question, and then we'll get to them at the end of today's presentation. All right, great. We've got a couple people that have acknowledged that everything's working, so that's good. So today's speaker is Dr. Cordelia Ziraldo. She's going to be reviewing Advaita's latest application, iVariantGuide, and discussing specific workflow concerning case and control variant analysis. Designed to help you quickly identify and visualize variants and understand how they may be affecting various systems. Cordelia is the product manager for iVariantGuide and is a computational biologist with expertise in systems biology approaches to understanding inflammation and immunology. Her graduate and postdoctoral research was focused in two areas. One, building mathematical models of biological systems to generate testable hypotheses, and two, generating biological insights from clinical samples using statistical and machine learning approaches. Dr. Ziraldo holds degrees from the University of Chicago and the University of Pittsburgh. At Advaita, she is leading the launch and development of the iVariantGuide platform and is excited to share the power of computational approaches with a wider community of biologists and clinicians. So with that, I'm going to turn it over to Cordelia, who's going to take you through today's presentation. Cordelia?
Thank you so much, Andrew. And welcome for attending. I'm very excited to show you how you can use iVariantGuide to get really all the way from a set of variants to biological interpretation. So the agenda for today, briefly, I'm going to introduce the company for those of you who are not familiar with us, and then I'm going to talk about what it means to do a case versus control variant analysis and the types of questions that we can answer. And then I will talk about how iVariantGuide is uniquely set up to address those questions. And lastly, I will demonstrate this with a live demonstration of a case study of a real data set. I'll go over the background of the data set and the results and what we've learned. And of course, we'll leave time for questions at the end. So Advaita BioInformatics is a company in Michigan. We have just under 10 employees, and we've been around for a few years. We're funded, we started out with funding through NIH, SBIR, and STTR grants. And we've been fortunate to make collaborations with a wide variety of top universities, as well as great collaborations and partnerships with other industry members. So Advaita's mission is to develop bioinformatics tools to simplify data analysis. We try to bridge the gap between data acquisition and interpretation. And we do that by using the most advanced computational methods coupled with a comprehensive knowledge base. In fact, we just updated our knowledge base last week, so all of the underlying data that you do your analysis on is the most up-to-date it can be. And we wrap those two things with user-centered design, which is one of the things that really helps us stand out from the crowd. We know that it is biologists and clinicians, life scientists who can have those really meaningful insights, and if only you can actually understand your data. So we try to take the math out of it and make it approachable so that the people who can make those insights and add knowledge can actually interact with the analysis. So I want to just quickly mention where we sit in the bioinformatics pipeline, just to orient you. This pipeline that I'm showing starts with measurement. So in the case of variant analysis, that would be a sequencing machine that outputs measurements, which then in the quantification phase are turned from intensities or other type of measurement into a biologically meaningful result, like a nucleotide, for example. There's also alignment and normalization and QA that goes into this step, so that at the end you end up with an entire sequence in the form of a FASTQ or FASTA file. The next step would be comparison, and in this case what we're talking about is calling variants. And beyond calling variants and generating a VCF, you might want to find variant sets by comparing multiple consensus sequence from different groups. I'll talk more about this as we go on. Where we come in is at the very end of this pipeline in the interpretation phase. So you take those variant sets in the form of a VCF file and upload them to iVariantGuide. And what we allow you to do is then annotate those variants with a wide array of domain knowledge and then prioritize them using dynamic filters so that you can find the variants that are most likely affecting the phenotypes you're interested in. We then allow you to go one step further and look at pathways, gene ontologies, and even related diseases. So a couple of benefits to highlight are custom filter presets, which allow you to take the same set of filters from one data set and apply it to another data set. This allows you to have a streamlined workflow. It also allows you to have provenance in your analysis. We also feature a printable report, which is a summary of your results generated automatically as a PDF, and online sharing, which allows you to send your interactive analysis to a colleague via email, making collaboration simple. Our entire platform is hosted on the cloud, which means you can access it from anywhere. You can do this analysis from your couch or your office, depending on the day. So let's talk a little bit about case versus control variant analysis and what you can actually get from it. The bottom line question really is, how can genetics explain differences between groups? Or, on the other hand, how can we use genetic data to explain similarities among members of a single group? And what I mean by this is several things. First, which variants are commonly found among samples from group A, but B? And on a next level, we can also look at what genes are commonly impacted by variants in group A, but not group B, or vice versa. And lastly, we can extract that gene information to a higher level and ask what mechanisms correlate with genes and variants associated with either group. So we're going to look at all three of those levels today. Pictorially, what we have are variants. When you compare your cases to controls, you're going to have variants that are only found in cases and variants that are only found in controls, and then there are going to be some variants that overlap. So your consensus variance is going to be based on some threshold side to set. It's going to be some mix of the case variants plus maybe a few of those variants that are. This is going to depend on your experimental design and a lot of different variables that are highly personal. So what I'm showing you here is that we're keeping all of the variants that are red and unique to the cases, but also a few that are blended between the cases and controls. And we're calling that our consensus variant set. So this is the variants we're going to move forward with into iVariantGuide in our analysis. The first step is to go from consensus variants to high priority variants. Not every single variant in that set of consensus variants is going to necessarily have an effect on our phenotype. So we do filtering in order to identify the ones that we think are most likely to be important. And then we go from high priority variants to impacted genes. And I'll talk more about how we do that soon. From impacted genes, we can then look at biological context and do this analysis of pathways and gene ontology terms to really start to probe mechanism of what's going on in this set of variants. So the first three steps are annotation and filtering, and that last step, moving into mechanism, which is really where iVariantGuide shines. So all of these steps, as I mentioned, are executed within iVariantGuide. Briefly, I'd like to just take you through what a workflow looks like in iVariantGuide so that you can be oriented to each of the modules. The first step is to add your samples or your VCF containing consensus variants by uploading your VCF to our web portal. And from there, you create a report. And this report is going to be the annotation of those variants that you just uploaded. You can do this using the user interface, or we now have an API client as well. The next step, once the report is created, is to filter your variants using our dynamic filters. I'll demonstrate this a little bit later. Each of our filters is also a snapshot of the statistics of the variants in your filtered set and allows you to see how changing one parameter can also influence others. And every single variant also comes with variant details, and we'll show you specifically which transcript changes are expected to happen, what type of effect the variant is predicted to have, whether there are any diseases related to this variant via ClinVar, and whether this variant is related to any pathways or ontology terms via the transcript that it impacts. Once you've set high priority variants, you can move on to pathway and gene ontology analysis, which are both highly visual and interactive. They allow you to see immediately where your variants are falling and also layer in other elements like microRNAs and drugs that might target the same genes where you have variants on the pathway. Similarly, gene ontology terms will show you the entire ancestor tree, and you can see how the variants in your set are propagated throughout. Lastly, I want to mention that we have a printable report. It's automatically generated PDF, makes it very easy to share the results of your analysis with your collaborators. This is a quick overview of the annotations that iVariantGuide provides. I'm not going to go through all of them for time, but what I want to say is that we go all the way from transcript level annotations up to phenotype level annotations. So we have sources from ClinVar and OMIM, as well as dbSNP, TargetScan. And I want to talk a little bit about SNPF, which is get our variant effect prediction from. In this case, a lot of other variant prediction algorithms use a continuous number system. So the higher your number, the more likely your variant is to be deleterious. But what SNPF does is it uses a discrete system and ranks each impact as high, moderate, low, or modifier. That's it. So high impact variants have a much greater probability of causing problems at the transcript level than modifier impact variants. I want to point this out because what I think it does is it makes it very simple to identify variants that could be deleterious, and it makes it even easier to get straight to your high priority variants. So, in addition to our graphical filtering, we offer presets, and I want to just quickly explain the benefit of presets. So, in this example, what I have is a sample with 34,000 variants, and I have set 3 different filter combinations. So, I've set a threshold for quality and minimum read depth. I've also removed any variants that occur in ClinVar and set the minor allele frequency extremely low. And then I've set the predicted impact from SNPF to high and moderate. So I want variants that I expect to have at least a moderate impact on the transcript. And what I've gotten with just a few clicks is from 34,000 variants all the way down to less than 300 variants. And then the next thing I did is I saved this combination as a preset. I call it novel deleterious mutations. And the next time I have a new sample, I can apply the same preset, and with only one click, I can get from, in this case, 110,000 variants all the way down to 371. Saves me time, and I'm sure I have the exact same settings, so those two sets of variants are now comparable to one another one-to-one. I want to quickly point out a few extra details about our pathway analysis. We provide correction factors, which allows you to really understand which pathways are statistically significant when you consider the number of genes impacted by variants. So which pathways are really enriched for variant impacting genes in your data set. We also show you this visualization, which gets you straight to mechanism. And then we overlay microRNAs and drugs, as I mentioned earlier. Our gene ontology analysis includes not only visualizations and corrections for multiple comparisons, but we also provide pruning factors, which allows you to take advantage of the inherent structure of the ontology in order to get more specific results that are more indicative of what processes and phenomena are really being impacted in your data. And lastly, the printable report includes pie charts and histograms of all of the different filters and annotations, regardless of whether you selected them or not, and top results for both pathways and gene ontologies, as well as methodology, including equations and references. So let's get to the meat of it. Our demo data set is RNA-seq of breast cancer subtypes. This was done RNA-seq using an Illumina HiSeq 2000 on mRNA from breast tumors from 3 different breast cancer subtypes, as well as normal tissue. So in total, we have 20 samples, and the 3 different subtypes are, TNBC, which is triple negative breast cancer, non-TNBC, and HER2 positive. I'll go over those in a little bit more detail on the next slide. But first I want to point out that when you're doing RNA-seq as opposed to DNA-seq, what you're looking at are variants in sequences that are actually being expressed. And that's important to keep in mind because our breast cancer subtypes are actually defined by gene expression patterns. So, triple negative breast cancer is defined as there is no expression of either HER2 or the estrogen receptor or the progesterone receptor. In contrast, non-TNBC, non-triple negative cells express either the estrogen receptor or the progesterone receptor or both. And that could be one of the 3 genes that I've listed here. And as you might expect, the HER2 positive samples express HER2, which is also called ERBB2. So let's talk about what I did in order to get to consensus variants. So using those 20 samples across all 3 subtypes plus the normal, I pooled all of the variants together, and then I removed any variant appearing in a normal sample. So I didn't want to have any overlap between variants appearing in normal samples and variants that I'm calling breast cancer. Once I removed the variants appearing in normal samples, I also removed additional variants that appear in all 17 breast cancer samples, because I expect that those are probably sequencing errors, and we can call them false positives. So what I'm left with now are variants that are appearing in no normal sample and in less than 17 breast cancer samples, so fewer than all. And the last thing I did was I said for each subtype, retain only the variants that are found in at least 2 samples. And that's what this yellow, blue, and green coding is here. I'll show you on the next slide what we end up with are approximately 50 to 75,000 subtype variants for each subtype. And some of those subtype variants are shared and some are unique. The next thing we did was analyze these sets of consensus variants in iVariantGuide. So the first thing we looked at were breast cancer predisposing genes. It's a list of 8 to 10 genes that are known to affect risk of breast cancer, and were listed in the introduction of the paper that this data comes from. And what we find, which is comforting, is that the only subtype variants that are occurring in ERBB2, which is HER2, happen to be HER2 subtype variants. So there are no triple negative subtype variants that affect ERBB2. There are no non-TNBC subtype variants that affect ERBB2. Similarly, for the progesterone receptor and ESR1, only non-TNBC subtype variants affect those genes. We also find, unsurprisingly, that BRCA2 is actually affected by variants from all 3 subtypes, whereas BRCA1 is only affected by variants from HER2 and non-TNBC. So let's look at what this looks like on an actual pathway. This is a pathway map that I downloaded and exported directly from my analysis in iVariantGuide. And what you see here are variants that are unique to non-TNBC. So in the top left corner, you'll see that I've shaded in green the region of the Venn diagram where the variants that are in this analysis lie. So these are variants that were only called subtype variants for non-TNBC. They were not called subtype variants for TNBC or HER2. And when we look at the breast cancer pathway, what we find is a ton of hits in this luminal A and luminal B section, which is exactly what non-TNBC is. So these are all variants in the estrogen receptor, as well as the progesterone receptor. And additionally, there are a few other variants in other cancer-related pathways. So here's a RAS variant, here's an integrin variant. Importantly, I want to point out that the preset that I used did not select for cancer genes. The only criteria I used were quality threshold greater than or equal to 10, and predicted impact of high, moderate, or low. So I didn't want to include any modifier variants. I only wanted ones that I thought would have a substantial impact on the transcript. So this is unique to non-TNBC. If we then look at the entire green circle and say, okay, fine, we can include things that overlap, but we really want to focus on non-TNBC. What we'll see now is a few extra things. So now, all of a sudden, we have a few more variants in PI3 kinase and AKT, as well as down here at the bottom, there's some new variants here as well. So we can do this type of analysis for all 3 subtypes, and I won't go through it in detail, but I will show you the consensus variant map for each of the 3 subtypes, and these colored circles will continue to indicate the regions where you have unique variants. So for example, for HER2, the shading actually contains all the variants on the breast cancer pathway in any HER2 subtype variant, but within the blue boxes, all of these variants are unique specifically to HER2 samples. Again, this little cluster here of APC and CK1 alpha and beta-catenin seems to pop up when we look at the overlap. And lastly, when we look at triple-negative variants on the breast cancer pathway, the only genes that are actually unique to triple-negative samples is this ERK1/2. However, you can see that there's more shading upstream of that ERK because there's other variants that overlap with other subtypes affecting this mechanism. So it's not surprising that we see effects of proliferation and survival in these samples. Putting it all together, you can see almost all of the mechanisms that are drawn on the breast cancer pathway are affected by one or more subtypes. But we also get a nice overview of where there might be overlap between the subtypes and what those differences are. What I want to do now is take you into the application and show you how you can get a result like this pretty quickly and what new things we might be able to learn. So, this is my reports page. The first report I want to look at is the intersection of all three sets of subtype variants. So this is going to be that center triangle of the Venn diagram. These are variants that are called subtype variants for all three subtypes. And I've applied a filter, which is a set of genes, that set of breast cancer predisposing genes that I talked about earlier. And the other criteria here are that I want the impact to be either high, moderate, or low. So you can see with our impact filter, though we've selected high, moderate, and low, the only resulting variants are all low impact. And here they are in the table. You can see that we went from 6,000 variants to 4 using this list of genes and this set of impact criteria. And here are our 4 variants in the table. And you can see that one of them is, 3 of them are clinically significant, and one of them is not benign. So we'll click 2, and what we have are details about this specific variant. So I'm going to zoom out just to the, no. And at the top we have the chromosome position and allele change, a link to, and talk about the changes at the transcript and protein level. This is a synonymous variant, which is why it's rated as low impact, but that doesn't mean that it's not necessarily having an effect. The next section is diseases, and as you can see, this variant is related to breast and ovarian cancer, as we all know. These little menu give us exact ClinVar record where you can go and review the evidence, as well as additional resources like MedGen and Orphanet. For rare diseases, we also, for genetic diseases, we also link to OMIM, as well as SNOMED and Gene Reviews. The next 2 sections are pathways and go terms, and these are going to be any pathways or go terms that are related to this variant via the gene it impacts. So that's the variant details view. But what we really want to look at are the pathways that are affected by these variants. So rather than select a set of genes, I'm going to apply that same preset that we were looking at in the PowerPoints. Over here in the left-hand pane, we have all of the presets available. I opened this little for impactful variants, and the first thing I'm going to do is click the I, which is for info. What that does is it pops up the preset information, so you can see what the settings are for that preset and make sure it's the one that you really are interested in. This matches what I said before, so impact selected high, moderate, or low. and quality greater than or equal to 10. So I'm happy with that. I'm going to go directly to the pathways analysis for that preset. At the top, what you can see are the number of variants remaining after this preset was applied. So we started with 6,000. With the previous preset of that list of genes, we were 4. With this preset, we have about 300. This isn't a better number for pathway analysis because we're more likely to have several genes that are impacted on a pathway. I'm going to apply correction factors in order to really understand which pathways are significant. And as you can see, the most significant pathway here is systemic lupus. So when I open up this pathway diagram, what we see is that there's several genes with moderate and low impact variants. This key here at the top tells you how many genes with high, moderate, low, and modifier variants are impacted by variants in the preset on the pathway. So here we have a couple of MHC2 genes as well as genes. So that means that there's probably some genetic regulation going on in these samples. Below we have the list of variants and the genes that they impact, as well as microRNAs and any possible drugs that target genes on the pathway. If you're interested in designing an experiment to test whether or not any of these variants are actually making an effect on your pathway, you could think about applying a microRNA to a certain targeted gene and testing whether or not knocking it down has a similar phenotype to the mutated version. So for example, the microRNA that I just selected hits this gene right here, so it's hard to say whether or not you would see an effect. But it also hits this gene here, which is in parallel with this HLA, as well as IL-10. So we would expect to see quite a bit of change in B-cell function. Let's go and look at the types of mechanisms that are actually specific to one of the subtypes. I'm going to go back out to my reports page, and I'm going to open to HER2. So these are going to be the variants that are only called HER2 subtype variants. They're not called subtypes of any other subtype. So what I want to look at actually here again are the impactful variants, double checking my info, and I'm going to look at Go terms because what I'm really interested in are the biological phenomena that are corresponding to the genes that are unique to this set of variants. So take that set of genes that's unique to this variant and say, what are the biological phenomena that correlate to these? And what I can do in order to get the most specific results is apply not just a correction factor, but a pruning factor. So Elam pruning gives me the most specific terms that are also significantly connected to that set of genes that are unique to HER2 variants. And as you can see, the ordering of the terms updated. And the first couple of terms, integral component of luminal side of endoplasmic reticulum membrane. And what we have here relates to this term. So as you can see, again, we have the number of genes impacted by variants that are selected by the preset that are annotated to the term. And in this case, we have one gene with a high impact variant, 4 genes with moderate, and 4 genes with low impact variants. Again, the variant list is below. And we also show you parents and children of the Go term that also might be significantly related to this set of variants. In this case, there are no child terms, meaning this is a this is a leaf node. This is the very end of the tree. So this is the most specific term that you could possibly see. But both of the parent terms directly above it are also very significant because they're annotated with, if not exactly the same, then a similar set. Below that, there are also references. So this allows you to explore further if this is of interest to you. I said I would show you the printable report, so I want to make sure that I do that. With a preset selected, you can print a report. And what this is a very comprehensive summary of your results, starting with an introduction and going through methods and results for all of the modules. So as you can see, this goes on for quite a while, including an overview of the sample variants, all of the settings for your preset, and then the results after filtering. So we'll show you every single filter. We'll show you not only the filtered variants, but also how that distribution compares to the sample variants, which tells you a little bit about how applying filters changed that distribution. And then we'll also show, here we are. When you actually have selected so here we know we selected impact of high, moderate, or low, and that's indicated with this asterisk here. As you can see, there are 0 modifier variants because we specifically excluded them. We go on to clinical significance, and then we get to pathway analysis. So again, this is really in-depth. It includes full methods with equations. There are references, and there's a full list of references at the end. And then we show you those top 5 most significant pathways and give you p-values that are corrected and uncorrected. And as you can see, marked in green when they're significant. In this case, all 5 were significant by all measures. And then we're getting to the good stuff. So here's that pathway map, and as you can see why antigen processing and presentation came up as our top pathway. This entire mechanism is impacted by variants. It makes perfect sense why you might see this coming up at the top, and that perhaps there's going to be some dysregulation there. So this is the phagosome pathway. Some of the same genes are overlapping. And here we go. So there's 5 of these. And then we get to gene ontology analysis. So we'll do this. The gene ontology results are broken down by the 3 root ontologies. So there's a set for significant biological processes, a set for significant cellular components, and a set for significant molecular functions. And again, we'll give you the top 5 results for each of those, with first just corrected and uncorrected for multiple comparisons, but also using those pruning methods that I was talking about. What are you being there? And what could the most specific mechanism be? So again, in this case, we're seeing antigen processing and presentation come up near the top because there are a lot of MHC genes impacted by variants in this particular data set. We'll show you the ancestor chart for each of those top farms and include the key so that you can see how those ontology terms are related to. And we do this, as I mentioned, for all three root ontologies. At the very end, we have references for you. And this is a comprehensive list of everything that was highlighted in the report. So this whole thing is about 25 pages. With your account, you can download an example printable report by opening the preset bar and clicking on the example report. And if you have a subscription, you can generate one of these for any combination of report and preset that you would like. You can also share your interactive analyses using the share button, and you can decide if you want to include a preset so that when your collaborator opens up the report, the variants are already pre-filtered for them. In this case, the default preset is impactful variants, but you could set it to that set of breast cancer-related genes, for example, simply by typing an email and clicking add. So now this person can receive this report, and I can control whether or not I would like them to be able to share it forward, or whether I want them to see and interact with my analysis, but I do not want them to be able to share with another person. And this way you can control who has access to your data. If you're publishing, we also provide a public link that would allow you to share the report with anyone reading your manuscript so that everyone can go in and interactively view the results that you generated. Okay. So I would like to summarize. I think the important take-home messages from this demonstration are that iVariantGuide is really uniquely suited to find and identify the mechanisms that connect a set of variants. Get to that biological mechanism and figure out what your story is. It's really easy to find, and the layers that we provide allow you to really tell a rich story and generate hypotheses. Our presets make it easy to compare data sets. As I mentioned, it means that you don't have to redo the same steps, which means that you're sure that you've done the same steps on two different data sets. And lastly, our cloud-based sharing and printable reports really make collaboration simple. We want you to be able to share your findings so that others can benefit from them. And the cloud-based sharing means that other people can interact with data that you've already analyzed and maybe find new insights that you didn't find yet with their own presets. And printable reports mean that you're really ready to go in terms of including your analysis results in any manuscript or presentation that you're ready to share. So with that, I want to thank you all and take any questions. You can input your questions into the section in the control panel, and if we don't get to your question, we will follow up after the webinar with a personal email. So please feel free to ask as many questions as you have, because we'll make sure to get answers to all of them. Okay, I also want to highlight a couple of new features and subscription options. So first of all, our API client is published, which means you can, with your subscription, you can also add on API access. So if you have a lot of VCF files that you want to analyze or share, then the API access might be something you're really interested in. Next, as I mentioned earlier, our Advaita knowledge base was just updated to v1702. This includes very up-to-date annotation sources for your variants. I also want to point out we have new subscription options, including an end-to-end solution. So while iVariantGuide sits at the very end of that bioinformatics pipeline, we actually now can offer sample to interpretation. So we'll help you get your samples sequenced, and then processed, and all the way up through interpretation in iVariantGuide. And very exciting, we are now offering subscriptions that are HIPAA compliant as well. So if you're working with clinical data, this might be something that you're interested in. Contact us for more information. And last but not least, we will be attending the annual meeting of the Association of Biomedical Resource Facilities in San Diego at the end of this month. So if you're there, please stop by and say hi.
Great. Thank you very much, Cordelia. That was an excellent presentation. Hopefully people got some real benefit out of it. We have a few questions that are coming in. And so let me just kind of address those. Oh, by the way, I forgot to mention at the introduction, we are recording this webinar, and we will provide a link to you afterwards. I think it takes about 24 hours for the link to go out, and you'll have access to the recordings if you wish to review the presentation by Cordelia here. One question was, what was the workflow that you used to pool the different samples?
Yeah, I forgot to mention that. I used BCF tools, which are a command line tool that's available for free and has extensive documentation, but commands in BCF tools in order to generate those consensus variants.
Great. Another question came in here. Some of the variants you showed are common in that the minor allele frequency is greater than 0.1. Why do you think these might be important?
So I didn't filter out high minor allele frequency variants in this particular preset because I wanted to include. I wanted to be very, very inclusive rather than exclusive. I wanted a large set of high priority variants. Sometimes what ends up happening is that you'll have, with a set of consensus variants, you'll have several different variants hitting the same gene across your set of samples. And I wanted to see the genes that were really hit by a lot of variants. So I included all allele frequencies for that reason. Also because sometimes a combination of hits can, even if they're both common variants, having the combination is less likely. And that combination can be detrimental as well.
Gotcha. Another question is, can I export some of these images independently, or do I have to do it through that printable summary that you showed?
No, absolutely you can, and in fact the pathway images that I used in the PowerPoint presentation were exported directly from iVariantGuide as PNG images, but you can also export a vector image as well. As the other images, every single interactive filter, those pie charts are all exportable, as well as the Go ancestor charts. And all of the variant tables are also downloadable in CSV form.
Okay, great. I guess it looks like that's the extent of our questions. I'll give a few seconds if there are any last fleeting questions that people may have. Just a reminder, it's completely free to establish an account with Advaita and iVariantGuide. You can even analyze your VCFs completely for free. The only drawback is that you can't export the images and you can't save presets on trial analyses. But please feel free to poke around. It should give you extensive insight as to what you can do. If you have any questions about it, please, through the application, there's a button there for you to give us feedback, or you can certainly send us an email at info@advaitabio.com, and either Cordelia or myself will get back in touch with you. Okay, I think that concludes today's webinar presentation. Thank you, everybody. We really appreciate your support and your interest in iVariantGuide. Have a great day.